screen recording will be for our lecture unit 1.4, a little bit of microbial history, just some people that played an impact in the development of microbiology. So let's see if we can get it to full screen. Okay. So usually this unit is assigned as homework, meaning that you'll be looking at the slides at home. You can use your textbook. The chapter 1 usually has a lot of historical figures in it, or the index. The people you'll need to know for our lecture exam 1 include van Leeuwenhoek, Robert Hooke, Red-Eye, Jenner, Virchow, Schleiden and Schwann, Semmelweiss, Lister, Darwin, Pasteur, Koch, Ehrlich, Snow, Nightingale, Alexander Fleming, Watson and Crick and Woese, and most of these folks will be mentioned in this PowerPoint. So first of all, looking way back in history, before microorganisms were discovered, we could call this as the age of speculation. Since microbes couldn't be seen and there weren't microscopes to observe them, basically microbes were unknown. Their processes were observed, however. For example, people could see the decomposition or rotting of food or plants, and certainly they could see and sometimes experience epidemics of infectious diseases. For example, the Black Death caused by the bacterial pathogen Yersinia pestis. And because people didn't know what caused these infectious diseases, often there would be people blaming other people, for example, calling people witches and causing these horrible infectious diseases, which cause even more suffering. A big discovery was the development of magnifying devices, microscopes. And although Anton van Leeuwenhoek, who was a Dutch linen merchant, probably was not the first person to develop a microscope, he's famous for he developed a very powerful microscope that perhaps had a total magnification of 200 to 300 diameters, and he was really curious. He loved to use his microscope to look at all kinds of materials, water and blood and sperm and plant materials and insect materials. And what made his observation so important is that he wrote them down, and eventually they were published by a scientific community in Britain, and thus they were spread worldwide. So it's van Leeuwenhoek who is renowned for being one of the fathers of microbiology. He's the first person to have observed and reported the existence of microbes, which he called his wee beasties, or animalcules. Another important person was Robert Hooke, who used a less powerful microscope, but Hooke made thin sections through the bark of cork trees and observed these little tiny, what he called cells. Now, one type of cell is a little room that a monk lives in a monastery. So Robert Hooke is famous for being the first person to use the term cell, which eventually becomes the basis for the cell theory of all living organisms. In the late 1700s, there was a country English doctor by the name of Edward Jenner. And Edward Jenner had survived smallpox himself as a child. Very often people would not survive smallpox. This is a viral infection. And in working in the countryside, Edward Jenner became aware that the country folks had made an observation that young women, often when they weren't married, they would end up milking cows, so they were called milkmaids. Often these milkmaids would become infected with a cattle disease known as cowpox. And cowpox it's actually a relative of the smallpox virus. It causes fluid-filled vesicles chock full of cowpox virus. But the country folks observed that milkmaids who had suffered and recovered from cowpox, whenever smallpox moved through a village or a town, those milkmaids would never get infected. And in fact, they could actually take care of family and friends who were infected with smallpox. So Jenner decided to run an experiment. He asked his gardener if he could inoculate the gardener's son with fluid from a cowpox blister. And here we have a painting. This is the milkmaid that has a cowpox vesicle on her hand. Edward Jenner's harvested some of the fluid which would have the cowpox virus in it, and now he's inoculating the poor gardener's son here. And subsequently then, Jenner challenged the boy with fluid from a smallpox vesicle. So um, what he did is we now call the process, he had vaccinated the child using the cowpox virus, and the immunity the child developed against the cowpox virus was able to protect the child from the smallpox virus. So Jenner repeated this experiment multiple times. Pasteur was the one that termed the process vaccination for vacca, from cow, the cowpox virus. And this is one of the, not the first, but certainly one of the safer ways to vaccinate people against smallpox. As our understanding of biology of living organisms grew, the cell theory grew, and this was developed by Schleiden, Schwann, and Virchow. And in the cell theory, we say that every organism is composed of one or more cells. So we talk about cellular organisms, organisms made of cells, and the cell is the smallest unit having all the properties of life. And then furthermore, Virchow added that all existing cells arise from pre-existing cells. This was the process called biogenesis. Life can only come from other life. Cells can only come from other cells. In contrast to abiogenesis, and abiogenesis, another way to describe it was the theory of spontaneous generation. And this was an idea held by many people that inanimate objects could turn into living organisms. Mud could turn into frogs. Flour could turn into little flour mealworms. Rotting meat could turn into flies. So a really good description of abiogenesis is here. We see in this link, this Bozeman Science link. So please, if you have time, take a look at that. And then another important foundation in biology is evolution and natural selection and the origins of life. So try to take a look at this second video. We then move on to Louis Pasteur, and Pasteur was truly a brilliant man. His accomplishments go on and on and on. So Pasteur is famous for he helped to disprove the theory of spontaneous generation of organisms. He helped to disprove the theory of spontaneous generation of microbes, helping to prove that cells can only come from other cells. So he helped disprove abiogenesis and prove biogenesis. He did important work in fermentation. He did experiments to show that microbes are everywhere. They're even in the air. He laid the foundation for the germ theory of disease, and the germ theory of disease states that a microbe, a germ, can invade another organism and cause harm, cause disease. He developed vaccines, including the first rabies vaccine. He developed a process of pasteurization in which liquids that are to be consumed are heated to kill unwanted microbes. This was Pasteur's experiment to disprove the theory of spontaneous generation. So in this experiment, he would take a glass flask and pour liquid into it, and then this is the formation of his beautiful, what they call swan neck flasks. He would heat the neck and then pull it out into this beautiful S shape. It looks kind of like the neck of a swan. And then the fluid, the broth, the soup would be sterilized, like kill all the microbes by heating here. And then he would put a cotton plug at this end of the swan neck flask. Now, the cotton plug still permitted air to flow in. So this was one thought. People thought that air was required for spontaneous generation. So in forming the swan neck flask, Pasteur permitted air to reach the sterile broth, but the microbes would be either filtered out by the cotton piece here, or because microbes, even though they're little, they have weight, they would settle in the crook here. So the microbes would never be able to reach the sterile broth. So then he let the broth cool, and as long as this neck was not broken, the liquid would remain sterile, clear. In fact, they're in the Pasteur Institute, they say they even have some of his original swan neck flasks, and I'm not sure if there's still liquid present that's still sterile. But this was to disprove that microbes don't arise spontaneously. And a lot of people thought that microbes could just arise um, from, for example, soup. And Pasteur was trying to prove no, microbes can only come from other microbes. And so in this second experiment, we see it's very similar here. The original steps are the same, but then what he would do is he would tip the flask so that the broth could come into contact with the microbes that had landed in the neck, and then he would write them up, and then eventually the microbes would grow, turning the soup or broth cloudy or turbid. In another experiment he ran is he performed the same steps here, and then he would just break off the neck of the swan neck flask, and then again the microbes could reach the broth, grow, the broth would become cloudy or turbid. But again, helping to prove that the microbes could only arise from other microbes. They couldn't spontaneously arrive just from the sterile broth itself. Pasteur was really important in saving the French wine industry. This was actually the origin of the process we call pasteurization. The French winemakers were panicked because many batches of their wine were turning sour, like acid, instead of the nice, beautiful flavor of the French wine with alcohol. So Pasteur did experiments to try to figure out, well, what converts the grape juice into the wine? So he ran a series of experiments. Here are his hypotheses. These are educated guesses to explain a phenomenon. So in his first experiment, his hypothesis was the grape juice spontaneously would ferment, making alcohol. So he boiled the grape juice to kill any microbes, any organisms present. He sealed the flask here so air couldn't get in. And what happened, the observation was there was no fermentation. The grape juice just remained grape juice, and there weren't any microbes present. So based on his observations, he rejected the first hypothesis that the grape juice spontaneously would ferment or spontaneously give rise to microbes. And then the second hypothesis was that the air somehow ferments the grape juice. So again, what Pasteur did, he basically repeated his swan neck flask experiment here. So here's the grape juice, he heated it, he drew out the neck so microbes couldn't get in, but air could get in. And again, he saw there was no fermentation. The grape juice just remained grape juice, and there was no microbes. So he rejected the second hypothesis. He disproved that air doesn't ferment the grape juice. So in the next experiment, the hypothesis was maybe bacteria ferment the grape juice into alcohol. So Pasteur sterilized the grape juice, and then he added bacteria, and I would love to find out which bacteria he added. It could have been lactic acid bacteria, which we have on our hands, for example, our mucous membranes. It could have been acetic acid bacteria, and acetic acid bacteria make vinegar, right? And so these bacteria then took the sugars from the grape juice and fermented them into acids. And the way we detect acids, high hydrogen ion concentration, is our chemoreceptors send signals that, oh, this is sour. So when Pasteur used the bacteria to ferment the grape juice, the bacteria carried out a fermentation pathway that produced acids, and thus the grape juice would taste sour. And so his conclusion was that bacteria could ferment the grape juice, but not into alcohol, but into acids. And this was what was happening to the French wine, is that the grape juice was being contaminated with bacteria, turning the grape sugars into acids rather than alcohol. And here we have, this is really our origins of pasteurization here. So in the fourth experiment, the hypothesis was that yeasts were the ones that would take the sugars from the grape juice and ferment them into alcohol. So here he took his grape juice, he sterilized it by heating it. So this is the pasteurization process, killing any unwanted microbes, any bacteria, and then purposely inoculated the grape juice with yeast. And we'll be talking about the Saccharomyces cerevisiae, often known as brewer's or baker's yeast. These yeast carry out alcoholic fermentation. They take the sugar, for example, from the grape juice, and through fermentation convert the sugar into carbon dioxide and ethanol, alcohol. So after inoculating the sterile grape juice, he sealed it, sealed the flask so that other microbes couldn't get in. And the result was the yeast fermented the grape juice, made alcohol, producing a nice, lovely French wine. And so the conclusion was that yeast fermented the grape sugar into alcohol. And again, this was the origins of pasteurization when we heat beverages, for example, milk, or heat foods to kill unwanted microorganisms. Ignaz Semmelweiss was truly a great scientist. He was a doctor in a Viennese hospital, and he was really concerned because in the pregnant women in the hospital, there were 2 wards for the pregnant women. One ward or one hospital, nurse midwives took care of the women that were in labor, delivering the babies. And in the other ward or other hospital, it was the medical doctors and medical students that would assist the women in delivering their babies. And what was appalling was that the women in the ward in which the medical students and doctors were helping with delivery of the babies, those women had much, much higher death rates than the women that were helped by the nurse midwives. And they were dying from a disease called puerperal fever or childbed fever. So Semmelweis was trying to figure out why this was happening and made the observation that normally in the mornings the doctors and medical students would do autopsies on patients who had died the previous day, the previous night. And then they would move from the autopsy room, they would then move to help their patients delivering their babies. And Semmelweis had the hypothesis that the doctors and medical students were carrying, we now know, microbial pathogens from their autopsies of the dead people. They were getting contaminated, their hands were getting contaminated with the microbial pathogens. And then when they went to assist the women in delivering their babies, they would be using their hands, helping to pull the baby out. They were actually inoculating, transmitting these microbial pathogens to the women. And as a result, they would end up with these horrible infections, this puerperal fever, childbed fever, and many of them would die because this was before we had antibiotics. So Semmelweis had the nerve to suggest this hypothesis and suggested that the medical students and doctors should be washing their hands in disinfectant before they examined their patients. And the doctors were incensed. Who is this Semmelweis telling us that we're dirty, that we're responsible for the deaths of our patients? And it's said that Semmelweis actually had a nervous breakdown. He was so harassed, and that he actually died from perhaps the same pathogen that caused the puerperal fever in the women. But his work did not go unnoticed, luckily. And we'll come back to the good results of Semmelweis's observations. So here we have the father of epidemiology called John Snow. This is about in the mid 1800s. This is not the Game of Thrones John Snow. And John Snow was concerned about an outbreak of a diarrheal disease in London called cholera. Now we now know that cholera is caused by a bacterium, Vibrio cholera, and we know the transmission is fecal-oral. People that have cholera, they shed the bacterium in their feces, and then wherever the feces goes, the bacterial pathogen goes. And so what John Snow did was he took a map of London and he mapped where the cholera cases were coming from. And we can see here these little red dots, these little red marks here on the map indicate where all the cholera cases were coming. So he was using mapping, trying to figure out, why are these cases clustered in this general area? And again, he wasn't sure what caused cholera. He wasn't sure how the people got infected. But one of his observations was that all of these people in this neighborhood, they all got their water from the Broad Street pump. So Snow came up with a hypothesis that there may have been something in the water from this particular pump. And I think maybe the pump was right there. That looks like Broad Street right there. Snow's hypothesis was that there was something in the water that when the people drank the water, then the people developed cholera. So he came up with the brilliant experiment of simply taking the handle off the Broad Street pump, so people could no longer get their water there. They had to go to other pumps in other neighborhoods. And what was remarkable was this helped stop that cholera outbreak. So again, Snow didn't know what was in the water causing the cholera, but he showed that it was the water itself. It was the water that the people were drinking that was leading to them developing cholera. So later it was determined that the source of the water for the Broad Street pump, it was fecal contaminated. It had feces in it, and that fecal material then would carry the Vibrio cholera from other people that had diarrhea. So it was kind of almost a feedback loop here. So Snow then is known as a father of epidemiology, and epidemiology is a study of epidemics in populations of organisms with the goal of trying to understand how the pathogens are transmitted, and what can be done to prevent the spread of infectious disease organisms. Florence Nightingale, back in the days, women weren't often involved in the sciences. They could be nurses, of course. And Nightingale, my understanding is she was really good at mathematics, and just like John Snow, she tried to use analysis to figure out what was killing people. So she was a nurse during some of the horrific wars back in the day. It's hard to imagine warfare without antibiotics. So her her observations led her to believe that a lot of the soldiers that were dying during the wars, it was not just from their injuries, but factors that contributed was poor food. So the soldiers were malnourished. They were living in absolutely filthy conditions. And so Nightingale used her understanding of how these factors of poor nutrition and filth would further lower the defenses of these injured soldiers. Um, one of the problems they were dealing with was an outbreak of typhus, which is a bacterial infection that's transferred by arthropod vectors. And so in making these observations, then she said the soldiers needed better food, they needed clean living quarters. She was able to greatly reduce the death rate of the soldiers. And down here's a little saying attributed to her: I attribute my success to this. I never give or took any excuses. So I think she would have been a hard taskmaster, and certainly she saved many lives. Another really important person in biology is Darwin. His famous book, The Origin of Species, describing biological evolution. And biological evolution had been described previously, but Darwin came up with one of the driving forces, like one of the mechanisms behind biological evolution, and that was natural selection. And again, we just always want to remember, as biologists, as microbiologists, what is biological evolution and natural selection? So the concept of natural selection is the environment chooses variants in a population of organisms that are best adapted to survive in that environment. And thus those variants that are best adapted, they'll survive longer, they'll have more offspring, they'll pass on their unique set of genes to their offspring. So consequently, over time, the genetic makeup of that population of organism changes, and as a result, the population becomes better adapted to their environment. So this change in the genetic makeup of a population of organisms over time is what we call biological evolution. And it's truly one of the more or most important driving forces in biology. And again, just this little saying from Charles Darwin, he's going to, it's not the strongest of the species that survive, nor the most intelligent, but the ones most responsive to change. So adaptability is crucial for success. Here we come to Joseph Lister, a tough Scottish physician. Lister had read of Pasteur's work showing that microbes are everywhere, even in the air. And fortunately, he'd also read of Ignaz Semmelweiss's work, Semmelweiss's thoughts that it was contaminated hands that were spreading the pathogens causing childbed fever. And Joseph Lister believed both Semmelweiss's and Pasteur's observations. Now, Lister worked in a hospital, and there was high death rates from people that had to undergo surgery or people that had traumatic injuries in which the skin was broken. And many of these people died from overwhelming infections. So Lister, using the work of Semmelweiss and Pasteur, started using a disinfectant, carbolic acid, a phenolic, as an antiseptic and disinfectant. So what he would do is he would soak bandages in it and use the bandages um, um, to wrap wounds in. The carbolic acid, the phenolic, would kill any contaminating microbes. He developed a little machine that would spray carbolic acid over the surgical site, the incision site during surgery, and then this again would help prevent contamination by airborne microbes or microbes on the hands or the clothes of the surgeons, as we can see in this sketch. And with these techniques, Lister was able to dramatically decrease the death rate of his patients. Many of his colleagues, many of the other physicians laughed at him, and said, oh, he's crazy, right? But the results were his patients had a much higher survival rate, and you can't argue with that. So we sometimes say that Lister was essential in developing surgical antisepsis. So antisepsis, we can think of it as a way to prevent infection. So if we take the suffix sepsis, sepsis is the presence of toxins or pathogenic microbes in blood or tissue. So Lister developed techniques to prevent the infection of his patients, antisepsis. Robert Koch was a German physician, and he and Pasteur were actually in a race to prove the germ theory of disease. So the germ theory of disease was that a microbe, a germ, could invade a person or an animal and cause disease. Now, some people thought, again, if we remember abiogenesis, even if some people thought that microbes could cause infectious disease, some people thought that microbes could arise spontaneously in a person. And a lot of times people thought it was because the people are of bad nature or they were evil or they did something to make God angry. So it was really, really important to prove that the pathogens causing infectious disease, they come from outside the host. They don't arise spontaneously inside the host. So to prove the germ theory of disease, Koch came up with 4 steps, 4 prerequisites, what we call Koch's postulates, to prove that a specific microbe causes a specific disease. And in these 4 steps, what was really important was the ability to isolate the suspect pathogen in pure culture. And this is where Koch's lab had a huge advantage over Pasteur's lab. Pasteur's lab was only using the liquid media we call broths, like soups. One of Koch's colleagues' wife had suggested that to use this as a solidifying agent to make solid media, that an extract of seaweed called agar be used. Agars were used to make jellies back in the day. So it was in Koch's lab that they first added agar to these liquid soups, these broths. And the agar, again, it's like a solidifying agent. It makes almost like a substance, almost like jello. And with these solid media, they were able to isolate microbial pathogens in what we call pure culture, just a single type of pathogen. We'll be doing this in the lab. You'll have lots of practice with this. But again, this was essential for Koch's lab to prove the germ theory of disease. And basically, the 4 postulates, the suspect pathogen has to be found in all cases of the disease of interest, right? You have to isolate the suspect pathogen in pure culture. And again, they were able to do this using their agar plates. And then the pathogen grown in pure culture must be inoculated into healthy test animals. And then the healthy test animals have to develop the infectious disease of interest. And the final step is, from those infected test animals, you must re-isolate the suspect pathogen, again in pure culture. And if you can fulfill all 4 of these steps, all 4 of these postulates, then you prove that suspect pathogen does indeed cause the infectious disease. And again, this is just the germ theory of disease. A microbe, a pathogen, can invade another organism, the host, and cause disease. We're almost at the end here. 2 people in the 1900s that played important roles in the evolution of antimicrobial drug therapy. Paul Ehrlich, described as the father of chemotherapy. Ehrlich developed the concept of finding what he called magic bullets, drugs that would specifically bind to pathogens within a human and cause harm to the pathogen without causing harm to the human host. One of these magic bullets he developed, this is not an antibiotic, but it is an antimicrobial, was called salvarsan. And it was an arsenic-based uh, antimicrobial. It was arsenic-based for the salvation from syphilis, thus salvarsan. And syphilis is caused by a bacterial pathogen, Treponema pallidum, which we will be discussing later. But arsenic is toxic all by itself, so this wasn't really a true magic bullet in that it caused harm to the human patient as well as to the bacterial pathogen. It was with the discovery of penicillin by Alexander Fleming in 1928 that we discovered the first true antibiotic. An antibiotic is a substance made by one living organism that will inhibit or kill another organism. And Alexander Fleming discovered that the mold, the fungus Penicillium, which we see a possible penicillium colony growing on an agar plate here, the penicillium secreted some substance, and initially Fleming didn't know what it was, would secrete a substance which could kill bacteria. Now these little dots on the agar plate out here, these could be colonies of the bacterium Staph aureus. And you'll see that surrounding the penicillium colony, there's a clear zone here. This is called the zone of inhibition. And Fleming guessed that the fungus was secreting some substance that was killing the Staph aureus. And so he thought, what if we could isolate that substance, which eventually was called penicillin, and if it wasn't toxic to humans, maybe we could use that penicillin to treat Staph aureus infections or other bacterial infections of humans. And indeed, with lots of work, took many people, penicillin was purified. It saved the lives of many, many people during World War II and in the following years. And that led to this great search for other antibiotics. In addition, Alexander Fleming also discovered the enzyme called lysozyme. Lysozyme is found in egg whites. It's found in our sweat, our tears. Our cells of our immune system make lysozyme. And lysozyme is part of our natural defenses against bacterial pathogens. We'll see lysozyme actually hydrolyzes an important component in the cell wall of bacteria called peptidoglycan. And I'll leave you with a couple of questions here. So any ideas what important discoveries Francis and Crick made? So they were the ones that described the structure of DNA, right? And then Carl Woese was a great scientist who used ribosomal RNA sequencing to determine the three great domains of cellular organisms: domain bacteria, domain archaea, and domain eukarya. All right, so I hope this will help you with your study guide questions. Look on Canvas. There should be a practice history quiz posted. There will be a few history questions on our first lecture exam.